Today we will discuss about these overexpression calreticulums, where they are expressed on the cells. So after we discuss these figures, we will understand that these overexpressed calreticulums are translocated to the cell membrane. In the previous video, we have understood that when the cells or melanocyte cells are under stress, under oxidative stress, they will cause overexpression of the calreticulum. However, in the previous video, we have not gone through the location of this overexpressed calreticulum. So in this figure, we will understand where these overexpressed calreticulums are translocated on the cells. So for this, they use the PAG1 cell lines, where they have introduced this oxidative stress and observe the calreticulum mRNA at different time intervals, and they can see that there is an increase in the calreticulum mRNA to some extent, that is till 1 hour. After that, the calreticulum mRNA goes down, and this one of the reasons could be that with increase in time, the oxidative stress is not enough for the cells to trigger the overexpression of calreticulum. And for this 30-minute, 1 hour and 2 hour, the increase in calreticulum is statistically significant compared to the control case. And in order to further validate this thing, they use the Western blot on this PAG1 cell line and also this mesochemical cell line, melanocyte cells obtained from the patients. And they can see that with increase in time, there is an increase in the calreticulum expression in both the cases, that is both in PIG1 cell lines, which is immortal melanocyte cell lines, and these melanocyte cell lines from the patients. The calreticulum signal increases with time. In order to further validate their results, they do the immunostaining of the control cells and also the cells that are under oxidative stress, and they can see that the green signals are higher in the case of this oxidative stress compared to the case of control, and green signals are representative here of this calreticulum chaperones. And one more thing that they have find out is that in the case of control, the green signals are within the cells, they are not reflected on the cell membrane. Whereas in the case of this calreticulum, they can see some of the signals that are marked by the arrow, that green signals are on the membrane. So it represents that calreticulum might be translocated on the membrane. And the quantification of these green signals is represented in this bottom side of this panel C, where they can see that fluorescence intensity of calreticulums is statistically significantly higher in the case of these cells that are under oxidative stress compared to the control case. So from this they made a point that in the case of cells, from this they reprove the point that they have discussed in the previous figure, that cells when they are under oxidative stress will have high calreticulum compared to the case of control cells. And in order to check whether calreticulum are expressed on the membrane, they did all the fluorocytometry analysis, where they can see that with increase in time, there is an increase in the cells that have calreticulum on the cell membrane. That can be seen in the flow cytometer quantitative results and also in the flow cytometers mean cell numbers. And after validating their results using flow cytometer, they did the Western blot using the different section of the cells. That is for the they did the Western blot for the membrane section and also for the cytoplasm. And as a control, for the membrane, they use this ATP1A1 as a loading control, and for the cytoplasm they use beta-actin as a loading control. And they can see that loading controls signals does not change significantly. However, for this calreticulum in the membrane will increase at different time intervals, indicating that calreticulum overexpression is increases with time on the membrane. However, for the cytoplasm, they do not see a significant increase and also the loading control, that is beta actin, also does not significantly increase. So both of this, not just both of this, all of these results together made a point that overexpressed calreticulums are translocated on the cell membrane and when the cells are under oxidative stress. So this is what this figure finally concludes. And if you need to understand more about their experimental details in detail, experimentals informations in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.